everyone, my name is Yo-Yo. Today I'll be presenting this work called a context-aware deconfounding autoencoder for robust prediction of personalized clinical drug response from cell-like compound screening. So the motivation of our work is firstly, the drug screening is limited to disease models, for example, cell lines. And sometimes the drug response on disease model does not reflect that of human. And deep learning is essential for the accurate prediction of patient-specific response to a new compound, and is also critical to personalized drug discovery. However, there are challenges that patient data are often too scarce, or existing methods are unreliable due to data heterogeneity or distribution shift. And there are often bias like batch effect and confounding factors. So our solution is we have developed a novel context-aware deconfounding autoencoder that can extract intrinsic biological signals masked by context-specific patterns and confounding factors. So here's the rationale of code AE. And you can see the source domain drug biomarker. They can be the cell line samples, and the target domain drug biomarker can be the patient samples. And usually the biomarkers are the drug target, and it's regulating gene interacting network. And the common drug biomarkers are their shared drug regulatory network. The private features for each domain are unique features that may contribute to drug response for each domain. Here's an illustration of coding method. So given the label cell drug response data, the aim of code AE is to predict individual patient clinical response, which the model hasn't seen before. And our model has 3 stages. I'll explain them one by one. The first stage is unsupervised pre-training, which the model takes the unlabeled data from both cell line samples and the patient samples and map them into a embedding space where the biological confounding factors are disentangled, and the distribution of the embeddings of the patient is aligned with the embeddings of the cell line data as so. And once we get the aligned common embeddings, it's further passed to the second stage. And the second stage is the supervised training for the fine-tuning, and we use the cell line samples as our label data to on the pre-trained model from the stage 1. And the last stage is the inference stage. The deconfounded and aligned embeddings of patient is first obtained from the pre-trained model, and then we use this fine-tuned model to get the final drug response of a given patient. Here is the base architecture of code AE. You see from the middle, there's a shared encoder which learns to map both cell line and tissue sample to extract the common intrinsic biological signals. And both of them have their own private encoders to get their private embeddings for each one of them. And there's a shared decoder which reconstructs the input sample through a concatenation of private and shared embeddings. And the reconstruction loss quality is measured by this reconstruction loss. And the private and shared embeddings are pushed apart through a soft subset orthogonality loss. And the shared encoder appended with an additional classification network, which will be further trained during the fine-tuning and perform inference during the testing phase. Our model has 2 variations, and they're majorly varied on the concatenation of the private and shared encoder. And we use 2 different loss functions. The first one is MMD loss. Another one is adversary loss in the form of min-max optimization between a critical network AIF and encoder components. Here's the result. We use the shared encoder from the pre-trained model to generate new representations for both cell line data and also the patient data, and just see how the alignment goes. It turns out you can see they're largely overlapped in this TSNE manifolds, which also shows our model does a good job in aligning cell line and the patient representations. Here's another result based on the experiment we performed to compare with the baseline models using the ex vivo drug response data from PTC. And our model is overall the best performer among all the baselines by both of the metrics AURC and AUPRC. And we further evaluate the model on another test to predicting the chemotherapy. And again, our model consistently outperformed baseline models in overranking on both AURC and AUPRC. And here is the result of the application of Code AE to personalize the medicine. We applied a spectral bioclustering to divide 59 drugs into 30 clusters from the predicted drug response matrix. And from this heat map, the darker, the stronger the response is. Among the 59 drugs tested, the top 3 most responding drugs are Gefinet, ACR, and the gemcitabine. They're also the top 3 predicted drugs based on the clinical study. And Gefinet is an EGFR tyrosine kinase inhibitor, and ACR can block the growth of cancer cells harboring the activated EGFR mutate. However, gemcitabine may not harbor the EGFR mutation. So, consistent with their with the drug mode of action, the predictions of the Gefinet and the ACR, they're similar, as you can see from this heat map, but different from gemcitabine, which is the only one that does not harbor the AGCR mutations. So to summarize the paper, we introduced the new transfer learning framework Code AE to predict individual patient drug response from a model trained from cell line data. And we also performed extensive benchmark studies which demonstrate the advantage of Code AE over state-of-the-art. The prediction of the drug response for patients are largely consistent with the existing clinical observations. And here are a couple of directions for the future work. Firstly, more sophisticated methods to integrate the data, and then maybe the interpretability of coding needs to be improved. And estimating the prediction uncertainty for each new case can further improve the performance of our model, and we think it's also critical for clinical applications. That's the end of my presentation. Thanks for everyone for listening. Thank you.